Bismillahirrahmanirrahim. I am Dr. Sara Khan, and today our topic of discussion is splanknology. So, first of all, you should know what is splanknology. It is the study of viscera and glands. What are visceras? The organs lined by the epithelium and underlying connective tissue consists of epithelium. The epithelium can be ectodermal, mesodermal or endodermal in origin. The organs which have epithelium of ectodermal origin, they are parotid, adenohypophysis and mammary gland. The mesodermal origin, gonads and kidney and heart. And the viscera which have the epithelium of endodermal origin, these are liver, urinary bladder, lungs, etc. The underlying connective tissue is mainly mesodermal, and it is mainly formed of intermediate mesoderm, lateral plate mesoderm and splanchnopleuric mesoderm. How you classify the viscera? They are classified, first classification is depending on the central cavity. For example, there are hollow viscera which have a central cavity. For example, stomach, intestine and ureter, and there are some solid viscera which don't have any cavity. For example, liver, spleen, kidney. And second is depending on coelomic coverings. Most of the organs in the body are covered with coelomic coverings. For example, heart is covered with pericardial. The heart is present in the pericardial cavity and is covered with pericardium. Lungs are covered with pleural membranes and are present in the pleural cavity, while the intra-abdominal organs are covered with peritoneal membrane and are present in the peritoneal cavity. But not all the organs in the abdomen are covered with peritoneum. Some of the organs which are not covered with peritoneum, and they are known as retroperitoneal organs. For example, the pancreas, ascending and descending duodenum, ascending and descending colon, and kidneys. ये थोड़ी सी मिस्टेक हो गई है बेटा. Pancreas, ascending and descending colon, and duodenum and kidneys. Now, glands. What are the glands? A gland is an organ that consists of specialized secretory cells. Secreted material is usually a liquid. They are epithelial in origin, present singly or in groups. They can be unicellular or multicellular, mainly two types, exocrine glands and endocrine glands. Here in this diagram, you can see that this is the surface epithelium. What happens? The surface epithelium invaginates into underlying connective tissue. This is the underlying connective tissue, and this epithelium invaginates in the underlying connective tissue and forms the glands. So, the distal part forms the secretory unit. This is the secretory unit, and this all is the glandular epithelium, and the proximal part forms the duct, and from the ducts the secretions goes on the surface. So this is the repetition which I have already told you. Development of glands. It develops as the invagination of the epithelium into the underlying vascular connective tissue. Distal part forms the glandular or secretory endpiece, which is functionally the active portion of the gland, and the proximal part form the excretory ducts, which opens on the surface of the epithelium in the exocrine glands. Some cells get detached from the epithelial surface, which are known as the ductless or endocrine glands. Here you can see that this is the surface epithelium, and the cells begin to invaginate in the underlying connective tissue. The distal part, this is the proximal part. Now this is the distal part differentiated into the secretory cells, and this is the proximal part connects secretory end piece with surface of the epithelium. In the case of endocrine glands, which are the ductless glands, the proximal part disappears. Here, the proximal part disappears, while this is the distal part forms islands of the secretory cells surrounded by the blood capillaries. These are the secretory cells and these are the blood capillaries. So, the endocrine glands are ductless glands and they pour their secretions directly into the blood capillaries. Now, what is the general architecture of an exocrine gland? It has a parenchyma and a stroma. The secretory part of any gland is known as parenchyma. It has secretory end piece and ducts. The ducts can be intralobular, interlobular, and the main excretory duct. And stroma is made up of the connective tissue. Okay, stroma is the connective tissue which is capsule, septa, and loose interlobar connective tissue supporting the parenchyma. It also divides the gland into lobes and lobules. Now, the classification of the glands. Classification based on site of secretion. For example, the exocrine glands. Exocrine glands pour their secretions with the help of their ducts. The endocrine glands are the ductless glands. Some glands are both exocrine and endocrine, and some are paracrine. Pour their secretions into the epithelial surface directly or through ducts. And endocrine glands are the ductless gland. Here you can see in this diagram, these are the exocrine glands, these are their ducts, and here you can see these are endocrine glands. They have no ducts, they are surrounded by the blood capillaries, so the secretions are poured directly into the blood vessels. So we have studied the exocrine glands and endocrine glands. Here comes the classification of exocrine glands. The first classification is based on number of cells. They can be unicellular or multicellular. Here it is, this one is the example of unicellular glands. For example, the goblet cells. These are the goblet cells which are present among other non-secretory cells. These are the goblet cells. For example, here in this diagram, these are the goblet cells and these are the tall columnar cells which are non-secreted, and mostly the goblet cells are present in the GI tract. And the multicellular glands formed by invaginations of the epithelial layer in deeper tissue to form a diverticulum. Proximal part forms the ducts and the distal part forms the secretory element. So, this is the first classification based on the number of cells. If there is single cell which is secreting the secretions, these are called the unicellular glands, while other glands, for example, salivary glands, parotid, submandibular, sublingual, pancreas, these are all the multicellular glands. Okay. Now we will see the classification of multicellular glands. Unicellular में we have seen there is the goblet cell which is a unicellular gland. In the multi, now we are studying the classification of multicellular exocrine glands. So, number one classification is based on branching pattern of the ducts. As I have already told you that any gland, the exocrine glands have a duct and a secretory part. Okay, so this is the classification based on branching pattern of the ducts. If there is no branching, these glands are called the simple glands, but if there is massive branching of the ducts, these are known as compound glands. For example, here in this diagram you can appreciate that here there is no branch. So this is a simple gland, and here you can see there are multiple ducts. Okay, this duct, these ducts take secretion from the secretory end, and then these ducts combine to form a single main duct which pours the secretions outside. Okay, so no branching, simple glands, and with branching, compound glands. Second classification is based on shape of secretory end piece. Okay, we have seen earlier that a gland has a proximal part which forms the duct and the distal part which forms the secretory end piece. So now what's the shape of the secretory end piece? It can be tubular like this. This is tubular. Okay, the shape can be alveolar or acinar. This is acinar, this is alveolar. Acinar means this is sac like, or it can be tubulo-alveolar, upward tubular and lower part is alveolar. Okay, tubular, alveolar or acinar and tubulo-alveolar. So if I say that a gland is simple tubular, so the simple means it has no branches, and tubular means the secretory part is tubular shape. But when I say compound tubular, it means that it has massive branches of the ducts and the distal part, secretory part is tubular. So further classification: simple glands and compound glands. Okay, now the simple glands, as I have already told you, they do not have the branches. Okay, without branches, they can be simple tubular further classified into straight simple tubular, coiled simple tubular and branch. Okay, simple alveolar and simple tubulo-alveolar. And in the compound tubular, compound alveolar and compound tubulo-alveolar. These are the simple glands. For example, simple tubular. This has no branches, and simple, this is the tube shape. So this is simple tubular. For example, mucous glands of the colon, intestinal glands or crypts of lubricant which are present in the intestine. These are the crypts of lubricant. Then branched tubular, these branching, and these are the tubular network. These are the glands in the uterus and the stomach. And this is coiled tubular, simple coiled tubular. For example, sweat glands. So simple tubular, simple branch tubular and simple coiled tubular. Then is simple acinar or simple alveolar. For example, small mucous glands along the urethra. Then branched acinar. For example, sebaceous glands of the skin. Again, as we have seen in the case of the simple glands, that we have divided simple into simple tubular, simple alveolar or simple acinar or simple tubulo-alveolar. Same is the case with compound glands. The compound glands can also be divided into compound alveolar, compound tubular and compound tubulo-alveolar. For example, this is a very good diagram. Simple tubular, crypts of lubricant, simple coiled tubular, sweat glands, simple branched tubular, fundic glands of the stomach, simple branched acinar, meibomian glands. Now here in this diagram, these are the compound glands. Here you can see what is this type. These are, you can see excessive branches, so you can say this is a compound gland. And when you see the shape of the secretory part, it is tubular, so it is compound tubular. Here in this case it is compound, and when you see the shape of the secretory part, it is tubulo-acinar or tubulo-alveolar. Okay, for example, submandibular gland, and the example of compound tubular is Brunner's gland. Okay, and this is compound acinar, and the example of compound acinar is mammary gland. Okay, we have seen the classification based upon the number of cells, the classification based upon the shape of the excretory part, and this is the third classification, which is based on the nature of secretions. The secretions can be mucus, they can be serous, and they can be mixed sero-mucus. So the glands can be mucus glands, the serous glands, or the mixed glands. For example, in the case of the serous glands, the secretions are thin, watery, and rich in enzymes and protein in nature. Their cytoplasm is granular, stained darkly from pink to dark purple, and rich in rough endoplasmic reticulum. For example, parotid gland and lacrimal gland. Here in this case, you see that the cells of the serous glands are pyramidal in shape and are very high. The lumen is very small. Here you can see that the nuclei are rounded and are towards the base. In the case of the mucus gland, the secretions are thick and viscid. And in this case, the cells of the mucus glands are somewhat flattened than the serous glands, and the nuclei are also flattened and more towards the base, and the lumen of the mucus glands is wider than the serous glands. What are the examples of the mucus glands? For example, the goblet cells or sublingual glands. In the case of mixed glands, they contain serous and mucus secretory units. Sometimes serous cells form these are the crescent-shaped caps, which are known as serous dominions. For example, submandibular gland is the mixed gland. This is very important classification, which is the classification based on manner of secretions. For example, the merocrine glands, apocrine glands, holocrine glands, and cytocrine glands. Okay. In the case of here in this diagram, the merocrine glands. In the merocrine glands, you can see here that these are the vesicles present in the apical surface of the cells, and the secretions here you can see in this cell, the secretions are poured into the lumen by exocytosis. Okay, through the process of exocytosis, the secretions are going away out of the cell. For example, pancreas, parotid gland, and protein component of the milk from the mammary gland. Now this is the apocrine gland. Apocrine gland, apex say apocrine. Okay, here in this case you can see the apex of the cell, this pinched off from the cell, pinched off apical portion of the cell. Okay, सेल का जो एपिकल पोर्शन है containing the secretions, it is pinched away from the cell. Okay, apical portion of the cell along with secretory product is pinched off. Okay, these are known as apocrine glands. In the case of holocrine glands, entire cell is shed along with secretory product, for example sebaceous gland. Here in this case, these are the whole cells, disintegrating cell and its released contents. As this case, whole cell is disintegrated. This is holocrine secretions. And here is another type which is known as cytocrine. In the case of cytocrine secretion, whole of the cell is secreted as secretion. For example, spermatozoans and spermatozoa and ovum from the release of ovum from the ovary. This is an example of cytocrine secretions. Okay, merocrine, apocrine, holocrine, they are examples, and cytocrine secretion. Here there is again a very beautiful diagram. This is apocrine, the apex is shed off. This is merocrine, the secretions are shedding in the secretions are poured by exocytosis. And these are the holocrine, in which whole of the cell is disintegrated from here. Whole cell is taking away the secretions. So this is all about today's lecture. Thank you.